Hello, my name is Nigel Curtis and I'm speaking to you today from Melbourne, from the Royal Children's Hospital in Australia. We're of course in the bottom right-hand corner of the world, and this map is the normal, of course, world map, and this one is distorted by the number of births each year. Of course, Australia reduces in size and other parts of the world increase. There are 130 million babies born each year, and the vast majority of those, 120 million, are born in parts of the world where TB is still endemic, and therefore they receive BCG vaccination at birth. And this is therefore the most widely used vaccine worldwide. Now, the reason why I'm speaking to you about BCG vaccine, you can see from this Wikipedia page, which tells you that the vaccine was invented by Professor Nigel Curtis when he was a medical student. Now, of course, this tells you more about how easy it is to edit a Wikipedia page than my microbiology skills. But this rumor has spread, and if you follow tuberculosis BCG vaccine on Instagram, you'll see this recent post that says, "So pleased that Professor Nigel Curtis invented BCG, #vaccine, #genius." Now, of course, this is fake news because the vaccine was created by Albert Calmette and Camille Guérin. And this was 100 years ago this year, and they created this by passaging Mycobacterium bovis 231 times over 13 years until it became avirulent. Now, after its creation, they distributed this vaccine worldwide, where it continued to be grown and passaged before it was first lyophilized in the 1950s. And during this time, it mutated. And we now know from more recent studies that different strains and substrains of BCG vaccine have important genetic differences. They're all missing the region of difference 1, RD1, which is what makes them avirulent, but there are many other genetic differences between these strains. And therefore, it's probably not a surprise that there's good evidence that different vaccine strains influence the immune response and potentially protection against tuberculosis. Now, the off-target or non-specific effects of BCG first came to my interest about 20 years ago from this paper that described the increased survival of low birth weight babies in Guinea-Bissau who were vaccinated with BCG at birth. And that difference in mortality cannot be explained by protection against TB alone, but in fact is explained by reduced deaths from infections other than TB, the kind of things that kill low birth weight babies in high mortality settings, such as pneumonia, sepsis, and diarrhea. Now, in this paper by Calmette from 1931, he described the dramatic effect that this vaccine had on preventing tuberculosis, but also mentioned a reduction in general mortality. And you can see from these figures here that there was a reduction in TB, but also there was an observed reduction in general mortality from illnesses other than tuberculosis. And so we now know that after you're given BCG, you'll have a specific immune response that protects you against the target pathogen, in this case, tuberculosis, of course. But there's also a non-specific immune response that protects against unrelated pathogens. This is sometimes called the off-target effects or the heterologous effects, or just the accidental advantages of BCG vaccine. And these off-target effects manifest as protection against non-tuberculous infections, perhaps allergic disease, autoimmune diseases, and malignancy. And so the overall effect of BCG vaccine is the combination of these. And so when we're looking at all-cause mortality or hospitalizations, we're looking at both specific and non-specific protection. Now, non-specific effects should not be confused with cross-protective effects. We know that BCG protects against the closely related Mycobacterium leprae, and so therefore it's a good vaccine to protect against leprosy. And in this systematic review by Petra Zimmerman in our group, she showed that there's also good evidence that BCG protects against cervical lymphadenitis and borreliosa, both caused by non-tuberculous mycobacteria. This is cross-protection against similar bacteria. Non-specific effects should also not be confused with downstream effects, such as when influenza vaccination reduces subsequent bacterial pneumonia, or that chickenpox vaccine reduces subsequent invasive group A strep disease, or that rotavirus vaccination, which has been associated with reduced febrile seizures and perhaps type 1 diabetes. But these are indirect protection from downstream effects. And also, non-specific effects should not be confused with simple adverse effects, of which we'll all be familiar, but also things like narcolepsy caused by one of the H1N1 vaccines. So returning to non-specific effects, observations in high mortality settings have been of great interest. And the first of these, and I emphasize high mortality settings, is that live vaccines, and in particular BCG and measles containing vaccines, are associated with reduced all-cause mortality. So BCG vaccine halves mortality from diseases other than TB, and measles containing vaccines halve mortality from diseases other than measles. And we'll come back to these other effects later. Now, the non-specific effects of vaccines are quite polarizing. People say they're for or against them, or they believe in them, or they don't believe them, or they're a skeptic or a non-skeptic. There's been a lot of debate and disagreement, and these polarized views lead to confirmation or refutation bias. So we tend to read those things that agree with our view. This causes confusion amongst people. This leads to a lot of hot air and discussion, high blood pressure, uncertainty, and uncertainty is a threat to vaccination programs, as of course we have all recently seen with COVID-19 vaccines. And I love this paper from Israel that I must confess to not understanding all these diagrams, but what it's really telling you is that we all tend to read those things that agree with our views, and to avoid reading those things that disagree with our views. And I think non-specific vaccines are highly susceptible to this confirmation and refutation bias. But of course, you're all scientists, you want to know what the evidence is. And I'm going to tell you that the topic is highly controversial and it's highly complex. It's not a simple thing. The devil is really in the detail. It's not a simple case of reading just the abstracts. And some aspects, particularly the fact that not all non-specific effects may be beneficial, is potentially unpalatable. So I'm going to have to give you, this is an enormous area, and so I'm going to have to give you a very quick tour with some detail to give you a hint of the controversies that are involved. I'll declare what I think right up in this review that we wrote relatively recently. We concluded that the non-specific effects of vaccines are certainly plausible and potentially important, but it's their clinical implications that are uncertain. And if you want to know more, you can also look at this recent review that I was involved with, headed by Nora Fritchie. So let's start with the fact that live vaccines reduce all-cause mortality. And I've already told you about the studies from West Africa showing, in trials showing reduced mortality in those who've been BCG vaccinated at birth, but there are many other observational studies showing reduced mortality in those who've been BCG vaccinated. And a WHO commissioned review back in 2014, but that was published in the British Medical Journal by Higgins et al. In 2016, concluded that the evidence suggests that the receipt of BCG and measles containing vaccines reduce overall mortality by more than would be expected through their effects on the diseases they prevent. That's I've already told you. But let's look at the evidence for that. They come from a number of different studies, observational studies and clinical trials. And in all these forest plots, they essentially show lower mortality to the left and higher mortality to the right. And I'm confining this just to clinical trials because observational studies are obviously more prone to bias than trials. But even clinical trials are at risk of bias. And you can see that only 2 of the clinical trials were deemed to be at low risk of bias. Both of these were in Guinea-Bissau. And the combined relative risk of 0.42 means there was a 48% reduction in mortality in those babies that received BCG. In other words, BCG Denmark in this case was associated with a halving of all-cause mortality. Subsequent to that review, there have been more studies from Guinea-Bissau. This one published in 2017, again showing the same effect with reduced mortality in those that are BCG vaccinated in a clinical trial. This one very nicely showing that whilst there was a dramatic reduction in infections, there was no change in non-infectious deaths, so a nice control group, if you like, and showing the causes of death in those infections in the higher in those who did not receive BCG early, basically sepsis, respiratory infections, etc. Now, there's also this study relatively recently that does not show an effect. Now, this study, one of the co-authors was Frank Chan, who's a proponent of the non-specific effects of BCG and other vaccines. This was done in 2 pediatric intensive care units in India. And in both of these units, there was no difference in mortality in those who were given BCG and those who were not given BCG at birth. and there was no effect, as I said, no effect whatsoever on all-cause mortality. Now, in this commentary, and I agree with everything it says, but mainly because I wrote it, I made the point that this was a good study. It was randomized and stratified appropriately. It was a sufficiently large study. There was high mortality, 16% of these babies died. BCG was given in the first 2 days of life, so early enough to have an effect. And they certainly did receive BCG because the vast majority had a scar. And there was an unambiguous outcome measure of death within 28 days. But the big difference is that this study used a different strain of BCG to all the previous studies that I've talked about already from Guinea-Bissau, which used BCG Denmark. This used the BCG Russia, and that may be an important difference, that maybe that not all strains of BCG have similar effects, both the specific and non-specific effects. And I've talked about the differences between strains. There is a large body of literature showing that BCG can protect against a variety of different viruses and bacteria and protozoa in animal models. And these were recently reviewed in this paper by Bridget Frame in my group. And my favorite study is this one in mice. And you can set up an experiment of murine malaria where after a week, you can arrange the experiment such that after a week, the parasitemia in these mice is nearly 100%. And when mice have 100% parasitemia, they all die. You can then get the same mice, or obviously not the same mice because the first mice are now dead, but you could certainly take the same strain of mice, this time vaccinate them with BCG, and now they're able to mount an immune response and fight that parasitemia, and they survive. And there are many similar experiments in many different animals using many different pathogenic challenges. What about humans? Well, there's a number of observational studies that show, for example, in this one, a very large study showing up to 37% lower risk of acute lower respiratory tract infection, very large study. Another very big study here showing reduced hospitalizations, both due to respiratory infection and sepsis. But these are not trials, these are observational studies. More recently, there's this lovely study by Sarah Prentice done in Uganda, published just a couple of months ago, which looked at infections in a randomized controlled trial, again showing reduced physician-diagnosed non-tuberculosis infections in those who received BCG vaccination at birth. There's also studies for other infections. There's an observational study here suggesting that BCG is associated with reduced malaria prevalence. Now, BCG is the immunomodulatory effects of BCG are routinely used for bladder cancer, but they're also associated with a reduction in the incidence of melanoma, leukemia, lymphoma, and one study suggesting may protect against lung cancer. And finally, there's a large body of literature showing a beneficial effect of BCG on autoimmune disease and allergy, and it's also been used for the treatment of type 1 diabetes and multiple sclerosis. Now, the question is what is the mechanism that underlies the non-specific effects of BCG? Well, there's been a lot of work on this, and I really don't have time in this short lecture to give you all the different theories, but they're nicely reviewed in this paper here from Nature Reviews Immunology by Helen Goodrich, and I encourage you to look at this if you're looking for a summary. But what we really now understand is in the last kind of 5 to 10 years, there's been an explosion in literature on the ability of BCG to induce trained immunity. And again, I would encourage you to look at this paper by my colleague Mihai Natia in Nature Reviews Immunology, which nicely summarizes how BCG through metabolic effects, which subsequently lead to epigenetic changes in cells in the bone marrow, that lead to enhanced immune response on subsequent challenge. So we now understand that BCG induces trained immunity, and this really improves the innate immune response. So what is it that leads to high blood pressure? Well, really, it's the second point here, that non-live vaccines, and in particular DTP, may increase all-cause mortality. And in that study that I've already mentioned by Higgins et al., having said that the evidence suggests that the receipt of BCG and measles-containing vaccines are associated with reduced mortality, they go on to say that receipt of DTP may be associated with an increase in all-cause mortality. Now, the evidence from this comes only from observational studies, because you will understand that it's very difficult, if not impossible, to do a randomized controlled study where you do not give infants DTP-containing vaccines. The forest plot's very similar, high mortality to the right. And you'll see that in these observational studies, there's a suggestion that DTP is associated with increased mortality with a relative risk of 1.38, but you'll see the 95% confidence intervals crossing 1. This paper was extremely controversial, and I encourage you to go to the BMJ website and read these 12 responses. They're very entertaining. It's a fantastic argument in the literature. Frank Chan made the point that he was one of the reviewers of this paper, and together with the other two had suggested that the study from Papua New Guinea should be removed because it was high risk of bias, and he should know because he had led that study many years before. That was one controversy, but the other controversy was in the finer points of the statistics, and the lead author, Julian Higgins, did agree that there needed to be a correction to the analysis, and that the relative risk, I haven't got time to explain the detail, but really with that correction, the relative risk was 1.53, and the 95% confidence intervals no longer crossed 1. Now, the controversy continued in the literature. Peter Arby's response to this review is summarized in this paper, and he had made this figure where he assigned each of the studies a risk of bias and showed that those that had a lower risk of bias were the ones that really showed increased mortality in those who received DTP. And you can see that if you just looked at those studies that were at low risk of bias, there was a much higher signal suggesting a detrimental effect of DTP. So you can imagine this has been controversial because it is seen as a threat to the vaccine program, in particular when there are comments like this, where Frank Chan, in one of those comments to the paper, wrote that if this is true, and that if DTP increases mortality by 38%, which is what is suggested if you just take those at low risk of bias, then this vaccine may be responsible for 310,000 deaths annually. So you can see that that is quite controversial, but I would highlight that all of this evidence comes from observational studies, which of course are subject to bias. But nonetheless, that's a very strong signal, and in my view, really should not be ignored. Now, the next observation is that non-specific effects last until a different vaccine is given, and a non-live vaccine may negate the beneficial effects of a previous live vaccine and vice versa. So if we look here, after receipt of BCG at birth, it may be that infants are protected until they get DTP, which may undo the beneficial effect of this live vaccine and make them more vulnerable and have a higher risk of mortality, and particularly females, until they get their next live vaccine, which is likely to be a measles-containing vaccine around 9 months. And if this was the case, you would expect to see lower mortality in the first period, a higher mortality after DTP, and a lower one after measles-containing vaccines. And that is exactly what you see in one of the figures in this systematic review. And you can see that each of these come from a single study, and they look at the relative risk of death after each of these vaccines. And you'll see there's, to the left, lower mortality after BCG, higher mortality after DTP, and lower mortality after a measles-containing vaccine. And that pattern is replicated again in all these different trials, with the one exception of the trial in Papua New Guinea, which really should have been removed. This effect is not confined only to high mortality settings. In this study from the USA, looking at very large database, showed that in over 300,000 children, that if the last vaccine given was a live vaccine compared with an inactivated vaccine, the risk of a non-target infection was essentially halved. Now, the final point to make about the non-specific effects of vaccines is that they are generally stronger in females and in the first few months after vaccination. Now, the sex difference is very nicely shown in this figure from Peter Arby's paper here that shows that the increased mortality in following DTP, the signal is stronger in females than males. Now, we of course shouldn't be surprised that there's a difference between the sexes. We know that there is difference in the immune response between males and females, as well as in the immune response to vaccines in general. And this is nicely reviewed in this paper here by my colleague, Katie Flanagan. There are many other factors, of course, that influence the immune response to vaccines, and this has been reviewed recently by Petra Zimmerman in my group. And of course, we've only had a time to talk today about a couple of those factors. So I've had to focus my talk due to the time just on BCG, but there is a mountain of evidence that shows the beneficial non-specific effects of measles-containing vaccines. And if I had another hour, I'd love to tell you about these, as well as the non-specific effects that are associated with other vaccines, such as the oral polio vaccine, the oral typhoid vaccine, and the RTSS malaria vaccine, for which it has been observed that there's increased mortality in girls following this vaccine. There's been a lot of discussion about that in the literature, which I would draw your attention to. And finally, influenza vaccine. Now, it's difficult to have a talk at this time without mentioning COVID-19. And it has been asked that, can the non-specific off-target effects of BCG and its ability to improve the innate immune response, can it be used to reduce the impact of coronavirus and perhaps other pandemics in the future? On clinicaltrials.gov, there are at least 17 trials now registered with varying different numbers of participants in a number of different countries. And one of these, the BRACE trial, I'm currently leading, where this is being started in Australia. We expanded to include recruitment in Europe and also in sites in South America. And in this randomized controlled trial, we are randomizing healthcare workers to either receive BCG vaccine or placebo. We're following them for 12 months, and during this time, we're asking them to, of course, have a respiratory swab for PCR to confirm COVID-19 with every time they get symptoms that might be suggestive, which of course we now understand is almost anything, and to register their symptoms on a custom-built smartphone app. We also have surveys every 3 months during the study, and we're collecting blood every 3 months, not just for serology for SARS-CoV-2, but also for complex deep immunological studies using systems vaccinology to understand the mechanisms that might underlie the off-target effects of this particular vaccine. So I'm going to end there, but I think there's a lot for us still to learn about the non-specific effects of vaccines, the beneficial effects, and also whether or not we might need to consider any rescheduling of vaccines to mitigate against any potential detrimental effects. But in the meantime, of course, the take-home message is that we could, of course, continue vaccinating all our children according to the EPI. So thank you very much for your attention, and I look forward to your questions. Thank you.